My homework question, if you like, was challenges and opportunities in large-scale biobanks. So I thought, first of all, I should define what we think about stratified, what actually stratified medicine means. So I looked it up on the Leeds University Stratified Medicine Hub, and their definition is very nice and succinct. It describes an approach in which specific clinical characteristics are used to improve the outcomes for the patient. I'd like to broaden that slightly in the context of this particular discussion and think about the role for large-scale biobanks, how they can impact obviously things around the treatment and prognostics and diagnosis of disease, but actually thinking further back and to demonstrate really how powerful these are as a research tool to understand the causative factors in disease and also health. I was at a meeting 2 weeks ago with Manir, Personalised Medicine Conference, and it was clear from that the information that can be fed in to this idea around stratified medicine or personalised medicine is of course very large. All of the omic technologies, genome, transcriptome, proteome and so on, and then things around lifestyle and family history. One of the best predictors of disease is family history, medical records and medical history and environmental factors. And thought about in that way, of course, large-scale biobanks are ideally set up as a tool in this field. So first, a quick bit of background on the environment. This is rather important. There are, of course, different types of large biobanks. Prospective studies, of which UK Biobank is one. They're generally large, well characterised, phenotypically at baseline when the people are assessed, with opportunities for enhancements. And I'll touch on that a little bit later. Excuse me, in very large studies, the only practical way of following up is by linking to their medical records with consent. And of course, it allows you to assess a very broad range of risk on a number of outcomes. You're not looking at a small number of risks in one disease. And of course, data are collected before the disease appears. Equally powerful are retrospective studies. They're generally smaller. They generally focus on a single disease or group of diseases. And of course, the phenotype there is highly validated before people are taken onto that study. And of course, the outcomes data are generally good. Clinical trials, of course, they are very, very highly controlled and phenotyped with good outcomes data. They are understandably focused on a single disease, but follow-up in that case may be limited. So this is rather hard to see. There should be a green box behind here. So these are the opportunities, if you like. Size, and I'll explain the benefits of that in a moment. I'll show you a few examples that we've used in Biobank of new methods of data collection, which are both objective and accurate and can be used at scale. Of course, we have the opportunity to collect biological samples from our individuals, process them and store them in a way that will allow a wide range of future assays, some of which we can't predict at the moment. We have the capacity to link to data increasingly. Of course, there are challenges around this. Anybody who's tried to do this with electronic medical records will know the challenge is, but we have the potential to link to electronic health records, and we are doing that. Importantly for us, we have consent for recontact, so users of the resource via us can recontact our participants and invite them to partake in additional studies. I mentioned the capacity that we have to enhance our study by doing additional measures. And as new technologies come on board, and as we've learned how to use them in this environment, of course, there is the opportunity for very large-scale data generation and using these new technologies at scale. Very briefly, I'll use, so focusing down now on UK Biobank, I'll use this as an exemplar. It's a study of 500,000 men and women in the UK, aged between 40 and 69 at recruitment. That was the only selection criterion. We ask extensive baseline questions, we take physical measures, and then we take a number of biological samples. And the important thing here, as I said, is that they're collected and processed in such a way that they will allow many different types of analysis in the future. We don't want to preclude the use of technology. Critically for us, and the fact it's embedded in the NHS, is we have consent from our participants for follow-up through all health-related records. So that's medical and other health-related records. And I mentioned this point about size. 500,000 people is sufficiently large for a number of people to develop a condition to allow reliable assessment of causes and interactions, of course, between different exposures to get those subtle gene-environment interactions. This table shows the expected numbers of incident disease, in other words, disease that occurs after recruitment during 5, 10 and 15 years follow-up. And actually, this was done on a fairly conservative basis. Of course, we can assess these numbers here. This was done on a fairly conservative basis, expecting the the worried well, the healthy responder effect. The average UK Biobank participant is about twice as healthy as an average member of the community. We can't confirm this number yet, but many of these others are actually very, very accurate. And you can see as you go out into 2 or 3 years from now and further out, we'll have very, very large numbers of incident cases of disease, which is a very powerful research resource. Just to make the point about size, why this is important, this is a slide showing the Prospective Studies cohort, which is about 1 million people. This is a study done in Oxford. It's a meta-analysis of about 1 million people, and it looks at the impact of 2 risk factors, systolic blood pressure along the bottom and age, stratified by age up here, and this is a risk score up the side. If you take this on 5,000 people, you can see that at best there's a sort of a left to right trend here with systolic blood pressure, but these error bars are so large it's very, very difficult to tease anything out of that at all. If you go to 50,000 people, you can start to see some pattern emerging from the noise. There is a general increasing trend of risk with increasing systolic blood pressure, and there appears to be an increasing risk of age. But even with these figures here, you can see apparently in the oldest age group, 80 to 89, this actually levels off and dips a bit at the higher blood pressures. And in the youngest group, 40 to 49, it appears to be J-shaped at the bottom. If you then go to 500,000 people, it comes into absolute crystal clear focus, and you can see this very, very clear association between age and systolic blood pressure and risk of a heart attack. It's very, very powerful demonstration of the value of size. I mentioned the baseline assessment. This is the information that we collect from our participants. They answered a great deal of, it provided a great deal of information on a touchscreen format, which of course allows you to code and capture the information very efficiently. It allows you to include things like assessments of cognitive function and hearing tests. Some of these data were enhanced in an interview with a nurse, and at the same time, repeat measures of blood pressure and vascular stiffness were collected. We did a very detailed assessment of eye health, everything from a logmar chart, the standard kind of chart you see in an optician, all the way through to a 3D image of the layers at the back of the retina. Some fairly standard physical measures, an assessment of physical fitness with a simple ECG, and then we collected the samples. And for most people, that was their last encounter. Since then, since completing the 500,000, we've been able to use new methods to enhance this and add data to our resource for researchers. This can be using pretty cheap and scalable devices like this device here. It's a wrist-worn accelerometer. It contains the same chip that measures acceleration and deceleration that you find in your car airbag, but in that case, obviously, it triggers the airbag. Here you are measuring movement in 3 directions. You get this apparently rather noisy data. These 3 colours here represent the acceleration and deceleration in the 3 planes. You can use this, people who are really expert in this area can derive specific sequences out of here, but as a covariate, over 7 days you can generate an assessment, a numerical value for physical activity, and also a distribution of the intensity of activity. And that's simply posted, people wear it for 7 days, they post it back to us, it's downloaded, the data are downloaded, all identifying data are cleaned off, it's recycled and sent back out again. And these things will go through about 50 cycles before they start to wear out. So a very cheap and effective way of doing it. We're using the web to collect data, so we're collecting data on cognitive function. Of course, this is vital. As you'll see, we're doing we're planning to do very, very large-scale imaging. Of course, by the time people present and are diagnosed with Alzheimer's disease, much of the damage is done. So if we can start to collect data on cognitive function before disease appears, this will be very powerful. We have information on diet. That's a repeated web-based instrument, so it's done over the year, 4 administrations over the year, and you can obviously see how people's diet changes. It will code into energy and also nutrients. We're assessing mental health and also occupational history over time. I'm causing havoc for this poor cameraman by moving around. And of course, we're now starting to look at mobile applications, the kind of applications you find on mobile telephones, either in the house or worn. The point about all of these things is they're affordable, you can do repeat administrations, and of course the efficiency of the data collection, coding and analysis is very high. I mentioned the advantage of building a biological sample resource. These are the samples that we collect at baseline. Other people will do different ones, and that's absolutely fine. The point about this is about 55 mils of blood. We collect a urine sample, a saliva sample, and a sample for future extraction of RNA. It's worth noting in this very, very large scale, clearly that the robustness of the data trail and the participant identity is very, very important to protect, and we can use these fairly standard, robust industrial methodologies to protect the identity of the individual. Follow-up. How do we actually follow people up? Well, I've mentioned this briefly. Again, it has to be affordable, repeat administration, and you've got this efficiency. And I touched on the dietary questionnaire, but the advantage of this is, of course, you can assess conditions that are very hard to find about through linking to the health records, whether that's primary care or hospitalisation. I've mentioned these here, and we're about to launch one around the quality of life. Data linkage. This might give the impression that this is quite an easy task. It's not, and anybody who's tried to do this will know, but we are making quite good progress now. All of our participants are registered with the GP. That's how we got their contact details. And of course, because the NHS provides the great majority of healthcare in the UK, we have a pretty broad picture of their healthcare. We have consent for recontact as well, so we can follow them up. But we have already linked to the register of deaths and the causes. We have cancer registrations, and those are refreshed and updated regularly. We have hospital data from England, Scotland and Wales. They're different data sets. And we have good coverage of primary care data in Wales and Scotland. And we are moving towards agreement with the three main providers of primary care data in England. But that is taking a little bit of time. But again, it's this point about it's comprehensive, it's scalable and affordable. I mentioned the point that we are doing enhancements. You can enhance using these remote devices, you can enhance using web. We're coming to the end of a pilot phase now of a large-scale multimodal imaging assessment, and the plan is to do this on about 100,000 of our participants. We're doing a pilot of about 6,000 participants in a single centre, and then we have to go back to our funders and demonstrate that it's feasible, and there are no issues with the process around incidental findings. This shows you a facility that we set up in our warehouse in Stockport. We have 2 MR stations, a 1.5 Tesla for a cardiac and abdominal assessment. We have a 3 Tesla machine that will do an assessment of the brain. We have an analysis of the bones and joints with a low energy X-ray analysis, a so-called IDEXA, and then an analysis of the carotid bifurcation using ultrasound. And at the same time, we repeat the baseline assessment. Of course, when you start to, the value of this is not only in linking this to other baseline data and other enhancements, but also the fact this is multimodal. There are many studies around that will do MR or other measures, but the fact that we've got multimodal measures in individuals makes it very, very powerful. So the last advantage really is that we can take the resource that we've got and start to use the technology and the methodologies that we have to generate very large-scale data resources. You can imagine processing 500,000 people, processing the blood from 500,000 people takes some fairly high throughput processes, and we can use these, now that we've finished the recruitment, to start to do useful work on the samples. And of course this gives you some advantages around quality and consistency of the way that you process and the data that you generate. It's more cost efficient because it's already in place, and of course it's very high throughput. And it actually allows for very efficient use of the resource. You're not repeatedly thawing and freezing samples and sending small aliquots out to researchers. You're using it once to do high data content assays. So to give some examples, we have genotyped the entire cohort in collaboration with the Wellcome Trust Centre for Human Genetics and Affymetrix, and we're just in the process now of completing imputation on those data. We're well into the analysis of a panel of about 40 biomarkers on the serum, urine, plasma and red blood cells of the entire 500,000 people. And we're planning for analysis of markers of infectious diseases again in the entire cohort. And we're turning our mind now to thinking about, well, how do we take this kind of approach and apply this high content screening approach to an analysis of the proteome and potentially the metabolome. There is one other advantage of this, and this slide demonstrates it. Actually, having these very, very large initiatives will galvanise the scientific community. We had a pretty short time to come up with a chip that was purpose designed for Biobank, and it was remarkable how a large group of academics really worked in a fantastic way to get this turned around and to get the contents of this chip agreed, ready for it to be manufactured by Affymetrix and then starting to be used. I don't propose to go through all of this, but there's a couple of points here. It has a very, there are lots of markers on here suitable for GWAS with a high imputation efficiency. We have rare coding variants in the exome, but importantly, I understand, reliable measurement of ApoE genotype has in the past been difficult using this technology, and a great deal of work was done by this group to optimize those markers to measure ApoE genotype. Just as an aside, interestingly, they recommended that we put some Neanderthal markers from our Neanderthal ancestors on there, on the basis that if they've persisted in the genome for long enough, they must have some purpose. Okay, so with opportunities, of course, come some challenges. And those challenges focus around the management of the resource, management of access to the resource, and of course keeping the quality up. That's absolutely vital. And then actually, I've made the point about data linkage as an opportunity. It brings with it challenges as well. And then management of the data, which I'll come on to. Just taking the first three first, they're really a question of management rather than anything more than that. Clearly, this is a very, very large undertaking, as are other biobanks, that is costly. Our principal funders have invested about 150 million in this over 12 years. It's very large and it needs management. And of course, making sure that all of the data and all of the samples and the facilities and the infrastructure is robust and resilient and secure is challenging. It's like running a small organisation. And of course, it's complex. When we were doing the baseline recruitment, we were recruiting about 700 people a day. And we had a staff of over 300 doing that. And that all need management. Now that we are open for use by the academic and industrial community, of course the access to the resource needs to be managed in a way which makes it as convenient and straightforward to the users as possible, but obviously complies with the terms of the consent and the ethics. Of course, if that was just one application, that would be fine, but we have multiple applications going at any one time. Of course, they're overlapping in the point that they're in, and each of them has to occasionally deals with some complex financial, and then of course we need to make sure that the material transfer agreements are dealt with properly. Management of the quality really is a kind of fundamental, that's the thing that keeps us awake at night, making sure that the quality of the data of the samples are as good as they can be. So it's the quality of the data and samples within the resource, but also making sure that the data that we get back from our research users are properly described. Thinking about challenges of data linkage and validation, well, we can link to these data, and I was chatting to Munir earlier, but they will be more or less descriptive of the disease that you're interested in. And typically in these kinds of studies, you will be looking at groups of 5,000 or 10,000 cases with 15,000 or 20,000 controls, and going through each of those medical records by hand is probably not feasible. So we have this kind of filtering effect. Starting off using the principal coded data, deaths, cancers, hospital data and so forth, and really working up, this is work led by Professor Kathy Sudlow, who's our chief scientist, automated algorithms to maximise positive predicted value, to kind of filter out those ones that you're quite sure about. You then go down this filter to look for additional coded data that may come from primary care or an enhanced cancer data set, disease registers, and these have more complex algorithms. But ultimately, you may get down to a smaller number which will need expert adjudication, confirmation and subclassification of the disease by a person. Now that can be done over the web. This is well-established technology, but you will still need a person to look at those. And this approach is being piloted and tested in a couple of disease areas, and we are making progress with this over the forthcoming year. Data management, now that really is a challenge. First of all, just straightforward and simply the storage volumes. If we go ahead and image 100,000 people using those multimodal approaches, our imaging data set will be about 500 terabases. The genotyping data itself is about 10 teradatabases, so that the measured data and the imputed data, I don't even know how to calculate what the whole genome sequence data size would be at 30-fold coverage on 500,000 people. So simply storing these data is a challenge. An even bigger challenge is making these data available. How do we do that? There are different ways of thinking this, but if we download all of these data, how do we actually do that? Do we send it to you on disks? Does it go through the academic network? And what if multiple people are applying for these data at the same time? Things move very, very fast in this arena. So we want to provide data to people on a fair and open basis. when the data, whether it's data that's generated in-house or come back to us, we need to make sure that we comply with international data standards and that we have appropriate metadata to describe these data. We may not be around in 10 years' time, but people may still be using these data. One of the few criteria of having access to this resource is that in due course you return your research data. Now again, that needs management because that information has to be checked, it needs to be put into the database, it needs to be properly described and made available to other researchers because that will be very, very valuable. How then do we go about mining this enormous data resource in a way which is useful for researchers? And what do we do about analysis tools? Do we act as, in effect, an escrow for analytical tools? Or are the analytical tools held outside by the researcher and they have access to a private area, or is data sent to them? And perhaps above all, data security. We really, really need to make sure that these data are secure. We have the consent of individuals to access their medical records, so we need to make sure that our data security is copper bottomed. So, in summary, large-scale biobanks, I think they are ideally positioned to support stratified and personalised medicine. Looking at the causes of disease and actually the causes of health. We can generate, if we go back to that slide of the human with all the different data sets around them, the generation of a personalised profile of individuals starting or undergoing treatment. We do, by definition, it's our job is to maintain secure, high-quality samples and data, but of course we have the infrastructure in place and the organisation to generate at least some of those data on a very large scale, cost efficiently and to high quality, to the quality which is demanded by the end use. So there are, of course, inevitably, and we really shouldn't underestimate these challenges around data management and investment in the data management structures at scale. This is only going to grow both in terms of size and complexity. A lot of these approaches, I wouldn't say we've answered absolutely everything, a lot of the approaches have been proven in UK Biobank and in other studies, and this overall high throughput approach, high quality approach, has been recognised by the opening of this facility in Milton Keynes, which is the NIHR National Biosample Centre, which has been set up specifically to provide biobanking and analytical services to the academic community in the UK. That's all I want to say. Just hopefully that tees up the day's discussion there, Munir.
Thank you very much, Tim. That was great.